and Renshaw Global Investor Conference. Our first presenting company this morning is Bioquark. Bioquark is a biopharmaceutical company dedicated to developing proprietary biological drugs for tissue regeneration and repair. And here to speak for the company is CEO Ira Pastor.
Good morning. My name is Ira Pastor. I'm the CEO of Bioquark, and we are a development stage biopharmaceutical company that is focused on biologic approaches to human tissue regeneration and repair. We are a company that was founded in 2008, incorporated in Delaware, and have our central labs in Tampa, Florida, as well as Philadelphia corporate offices. We're a young company, both operationally and financially. We've raised about $750,000 from high net worth accredited individuals. We're in the process of raising an additional $1 million right now via Reg D offering, to be followed by a $20 million cumulative direct, with the goal of providing the company with enough capital over the next 3 to 4 years to complete preclinical development of our lead biologic known as BQA, as well as conduct a series of clinical studies demonstrating an inducible endogenous regeneration event in our initial target organ system, which is the human kidney, which we think will provide a major value inflection within 3 years. We intend also to continue to capitalize on other tangential opportunities of the portfolio, specifically in the area of dermapharma and companion animal health. We start off with a simple number. OECD estimates by the end of 2012 around the world we're going to surpass $6 trillion in total healthcare expenditure. And though you can slice and dice that number a myriad of ways, for us at the end of the day that money trickles down to two things. Diseases that either have an underlying cellular degeneration or an underlying cellular damage component. That is where your cells are being destroyed and not being replaced, or where they're damaged to some level, genetically, epigenetically, and no longer function correctly anymore. And we as humans are notoriously bad at dealing with these things. We have little to no regenerative capability in our critical three-dimensional organs and tissues. And while we do have inherent DNA repair capabilities in our cells, which constantly surveil our genome, correcting daily damage, these capabilities are diminished substantially over time as we age. We rely on tools like organ transplantation and replacement, which, aside from coming with major problems like the host-first graft reaction and impact on quality of life, also have this horrible problem with regard to the donor gap that exists. And although this data is a couple of years old now, the trend is the same. The majority of all patients that require a new organ are going to die before they ever get on a list to get one. Pharmaceuticals have done an okay job for us in sort of slowing our downhill progression, but can do virtually nothing to actually reverse damage once it's occurred. And lastly, we have this evolving area of stem cells, which has generated a tremendous amount of excitement in the last decade and a half in the popular press, but which has generated surprisingly few therapeutic regenerative outcomes to date. And we just take a look at sort of the laundry list of technical and strategic issues involved in turning the cell into a therapeutic product. It is in our opinion that niche products will be more relegated to niche opportunities moving forward, and a lot less as the sort of major game changers that they're held out to be. So what's an alternative to all this? Well, we think we have a pretty interesting one. There are many species in the biological world that are excellent at repairing and regenerating themselves. Most of you are undoubtedly aware that members of the amphibian kingdom, including many species of salamander, can regenerate major portions of their tail and spinal cord in a matter of weeks in both structure and function. This holds true for their limbs. Here you're looking at the lens of an eye of a newt that's been completely cut out only to start to regenerate in a matter of days. That's a brain of a xenopus tadpole that has been put through a blender only to reform in 2 weeks. And while not as well known as their regenerative prowess, these species have exceptional capabilities for cellular repair. Cancer, as an example, is a phenomenon that is extremely rare in regenerative organisms. And even under the presence of potent carcinogens, when tumors do form, like that epithelioma, it is not uncommon for them to spontaneously remodel and become part of the normal tissue architecture. And as fascinating as all that is, what's most fascinating is the underlying process behind it all is one and the same. It's known as epimorphosis, and I'm going to briefly walk you through it here just as we begin to make some parallels to mammals in the following slides. So, here's our salamander whose tail's been amputated. At this point in time, the cells that remain enter what can be best described as a biological time machine of sorts, where they are spun back to an earlier stem cell-like, progenitor cell-like state, proliferate in a very large quantity, and then redifferentiate along a variety of different cell lineages. Now, of course, a tail is a complex appendage. It's made up of many different tissues. I'm just showing you nerves here for case in point. At this point in time, we exit a state of active cellular reprogramming and enter that of active tissue organization and morphogenesis, where those new tissues have to integrate with their appropriate neighbors. They need to be organized up along appropriate tissue, organ, and positional specificity. And at the end of it, the whole process needs to stop. And these organisms can do this again and again throughout their lifetime, perfectly in both structure and function. It would be really great if we could do something like this as humans. And the truth is we can. There's one problem, though. The period of time that we can do this is extremely short, occurring in the moments following fertilization when our germ cells, our egg and sperm, first come together. And then the capability is pretty much gone. It is the biochemical events that occur in the oocyte at this period of time, the opening of the chromatin, racing of epigenetics, DNA repair, lockdown of maternal genome, activation of embryonic genome, and so forth, that ensure that all our children were born age 0. They all came out kicking with 10 fingers and toes and arms and legs. And for the most part, we're not born with your traditional chronic diseases of older age. Children don't come out of the womb with Alzheimer's and Parkinson's and congestive heart failure. Something else is very important. And it deals less with the biochemical events that occur in the egg and more with the downstream microenvironment that results from it. The actively morphogenic microenvironment, whether we're talking about an embryo, a limb blastema, similar dynamics in the plant kingdom, that environment is exceptionally good at organizing in the tissue it needs and organizing out what it doesn't want. You can throw in all sorts of cellular junk into these microenvironments. Undifferentiated stem cells, differentiated somatic tissue, cancer cells. And you don't get a mess like that at the end of the day. You get a normal fetus and a normal limb. And this is extremely important because when we begin to address this whole area of regeneration and repair, not from a straight cell perspective, but from a cell microenvironment perspective, it allows us, in addition to address regenerative and repair outcomes, to also address a lot of the problems associated with the space. It allows us to address the double-edged sword of overproliferation and tumor formation. It allows us to address some of the unmentionables that the stem cell industry doesn't like to talk about, like size control, shape, positional identity. And finally, it allows us to tap into some of the natural self-correcting mechanisms that exist in cells, like somatic reversion, like aneuploidy correction. And look at them now, not just from an observational perspective, but from a potential therapeutic angle. So all that being said, what are we doing at Bioquark? We asked the question, as ooplasm-based reprogramming remodeling is something that's been studied now for 50 years in the Petri dish in the form of in vitro fertilization and cloning, and more recently, cell-free extract reconstitution experiments. We said, could we use the tools that exist now in the year 2012, like proteomics, like transcriptomics, and export that biochemical information in a standardized format, in the form of a biologic, to be used in mammals as a regenerative and reparative therapy. And that is what we call BQA, which is the lead candidate in our portfolio. And to wrap your head about it a little bit, this is not a traditional new chemical entity or a purified protein. And you have to think of what this product is from a composition perspective, a lot more like some of the heterogeneous byproducts that exist in the pharmaceutical market today, very quietly generating billions of dollars. So for the last few years, we've been running a variety of studies in our lab in Tampa, both in vitro and in vivo, trying to understand the global reprogramming and remodeling dynamics as they occur. and have studied a variety of models. And I'm going to walk you through some of the top line of what we see. We've studied BL6 metastatic melanoma model, intratumoral administration. You're looking here at the hind paw of a mouse, 3 weeks of administration. We see substantial reduction in tumor size, as well as reduction in expression in tumor invasiveness biomarkers. In addition to this, we monitored these animals for 3 months following these experiments that lived very well with no evidence of proliferation following this event until they were eventually sacrificed for further studies. All control animals died very quickly. We studied standardized 200-gram weight drop model of traumatic brain injury, intraperitoneal administration, where we saw substantial differences in gross brain anatomy presentation, as well as reduction in beta amyloid expression. I apologize for the quality of that slide, which is interesting in its own right, because it not only is beta amyloid hyperexpressed in brain injury patients, which is somehow connected to loss in memory, but potentially allows us also to address other diseases with an amyloid component, like Alzheimer's, like type 2 diabetes. These animals also showed substantial improvement in spatial learning and memory via Morse water-maze testing. We conducted a topical administration in a squalene monohydroperoxide-induced wrinkling model, as well as sub-Q administration in a model of induced alopecia. And lastly, we studied the material for long-term administration in normal, healthy animals. And while this was not a formal safety study by any means, we observed over long-term administration no evidence of growths, abscesses, immune response, behavioral changes. But most interestingly, the animals on drug for that period of time lived on average of 70% longer than the control group. This is an interesting data point to keep in our back pocket for a little bit down the line. And the fact that the only biologically immortal organisms that exist on this planet today, certain jellyfish and hydras and what have you, achieve this by wholesale tissue reprogramming organization. So coming back to the current day, as mentioned earlier, we have a 3 to 4 year goal to get through preclinical development and into clinical development in the human kidney. The human kidney is a huge area in the United States alone, substantial unmet medical need, and it's fairly uncluttered with any competition. $30 billion annually is spent combining dialysis and kidney transplant and rejection drugs. And we feel if we can show an endogenous regeneration and repair event in a patient and keep them from moving in either of those directions, that alone is a game changer. We've chosen to focus on an orphan niche indication of FSGS, or focal segmental glomerulosclerosis, which is a chronic degenerative disease of the kidney where a majority of patients end up in the first year urinating out their kidneys, and by year 3, 50% of them have to go on dialysis or get a transplant. And it will allow for us to have a rapid proof of clinical concept, as well as potentially take advantage of potential fast-track opportunities. We have an aggressive yet disciplined approach to get into the clinic with it by year 3, with single maximum ascending dose studies, in tangent with a 3-month tox, and ultimately starting in year 4, a larger phase 2 proof of concept. And then at that point, we can make a decision whether we continue development in the kidney in a more pivotal trial, or license out the kidney opportunity in a organ-specific vertical manner to a specialty pharma group that specializes in the kidney, and then move on to the next organ system of interest, whether it be the pancreas or the spinal cord or the heart. All along, keeping in mind that there are indeed tangential opportunities for the short term in dermapharma and companion animal health that we will be generating data for along the sides that could generate even shorter term revenues. We have a very active intellectual property program at the company from a variety of different angles, with 1 issued patent, 2 filings. And while we continue to proceed down these paths, the product, by its very nature, being not only a biologic, but also heterogeneous biologic, comes with some very interesting built-in protections, which we refer to as the Premarin bioequivalency barrier. But if a product like Premarin is in an account, has been on the product market for 70 years now and has never had a generic competitor, so we think a product like this yields some very unique barriers that make generic competition down the line a potentially obsolete thing. And lastly, this just sums up a bit of the story. We have regeneration and repair, which is a huge unmet medical need on all disease fronts. Transplantation, drugs, and stem cells are leaving a lot on the table today. And we feel what we offer in a single biologic is really a dual solution for addressing both of these types of needs, and allows us to do it in the format of a traditional biologic, both from an administration perspective as well as a regulatory perspective. And with that, I'll say thank you and be glad to open the floor to any questions.
Could you explain a little bit more about, I mean, the immediate seems to be animal and dermo-cosmetic, but yet the real thrust where you're going is on the organ side. So can you explain a little bit about how the dermo-cosmetic fits and the animal fits into what you're trying to do?
Well, along the lines of the kidney program, we'll be generating data in larger animals. And that data alone, based on the fact that kidney damage is one of the main killers of our dogs and cats also, is potentially something that we could, in a partnering way, talk to companion animal health companies about. Typically, those companies don't like to develop new stuff unless it's truly, I mean, usually they retest antibiotics and other types of substance vaccines and so forth. But we think something of this nature with that type of data could appeal to a player in that space, especially just anecdotally talking to folks that are pet lovers and will do anything and spend a large amount of money trying to keep those animals alive. Diabetes, kidney failure, cancer, these all kill our pets just like they kill us, and potentially yield some good opportunities. From the derma angle, we've generated data in those two models of wrinkling and hair loss. If there is a way to create a Botox alternative, I mean, Botox is a huge market nowadays, and if there is something that can reverse that damage as opposed to paralyzing your nerves, I think that might be pretty appealing in that space as well.
With the technology that you're developing, how far out do you think it's going to be from a timeline to try to get it to market and monetize?
We think the FSGS indication could be in a pivotal regulatory study 2014, 2015. We could monetize it earlier if we send it along the lines of an organ-specific licensing arrangement with a Baxter as an example that would take the kidney, run with that, and we can then go deal with the pancreas next or what have you. But we think there's a lot of ways to peel off components of the story and monetize them earlier than taking it all the way through ourselves.
Can you formulate this as a topical that will go through the dermis?
We've only done that in one model in a very crude liposomal vehicle, and we've not. Obviously, there are going to be the delivery vehicle is going to be crucial in administration and delivery of this material in all fronts, but we have not done active study beyond just a very crude topical delivery vehicle at this point in that one model of the swelling. I think it's very clear. Thank you.